InSpectr, a tool for multimodal visualization and analysis of spectral data. InSpectr employs voxel spectrum probing. When the user is hovering a mouse cursor over the 2D slice view, the spectrum for a voxel under the cursor is displayed in red. It enables fast and uninterrupted probing voxels spectra and comparing them between each other. 4 specimen regions are explored. The regions contain solutions of gold, barium, gadolinium, and a mixture of these elements. Spectrum probing allows the user to quickly identify energy peaks of different chemical elements and their locations. Another analysis technique is the selection in the spectral domain. The user can specify one or more rectangular filter regions in the spectrum view. The voxels with spectra that pass through any specified rectangular region are selected. The voxels, selected by the given filter, are highlighted in the 2D slicer and in the 3D renderer. Selection in the spectral domain allows the user to quickly identify areas corresponding to energy peaks or to other regions of the interest in the spectrum. We present a method for defining a color transfer function directly on the spectrum. The user defines a transfer function by assigning colors to energy values in the spectral view. The color of each voxel is determined by combining the energy response in the spectrum with the color assigned to that energy value. When the transfer function is properly defined, it is possible to visually distinguish regions with different chemical compositions by their colors. Spectral magic lenses show the spectral color image on the 2D slice. There are 3 possible operating modes. An overlay mode allows the user to alternatively view the scalar and the spectral data. An offset mode enables concurrent viewing of scalar and spectral data for better comparability. Split mode can be used if close proximity between scalar and spectral data is required for the comparison. When the user is hovering a mouse cursor over the 2D slice view, InSpectr visualizes the quantitative chemical composition via a pie chart view for a selected voxel. Alternatively, visualization of a voxel's element concentrations is available with a periodic table view. The concentrations of chemical elements are shown using colored bars in corresponding element cells. The higher the concentration of an element, the higher will be the corresponding bar. We utilize linked 3D element maps for the composition analysis. Element maps show individual chemical element distributions. They are shown in separate 3D views using volume rendering. The 3D view with computed tomography data provides a high-resolution context and is used for user interactions. Pie chart glyphs are used to locally probe elemental compositions. The glyphs are overlaid over the computed tomography data. Each glyph shows the average concentration of elements for the region beneath it. The remaining video shows how resolution, opacity, and spacing of glyphs can be varied to support different analysis goals. Thank you for watching. Goodbye.